Now the third part is about data visualization. And we're going to use those other libraries that I mentioned, Seaborn and Matplotlib, to visualize some of the data that we worked with so far. The two libraries, the two data visualization libraries, Matplotlib and Seaborn, are one of the leading libraries of like Python libraries that are used for visualizing data, creating figures from data tables. And there are others, for example, Bokeh, and you're probably familiar with some of those other libraries. So this is just two leading ones that are used widely, but there are others. Also, I know that many of you are familiar with R, and there are different libraries that are used in R. Some of those libraries also across R, Python, they have support for both. Let's start with these two. So first thing is to actually install libraries, and I assume that you already did that because when we did the import, it would have complained if you didn't have those. If you type pip install again, in general, it will tell you either it's satisfied or it will upgrade. So there is also a pip install update flag command that will enable you to upgrade your libraries. The first part is that import, which we already went over. And then what Ido created is a histogram of the length of the gene sets from the two gene set libraries. So as I mentioned, in those gene sets, because we set up different thresholds, or have a threshold that included different sets of genes. For the CCLE, there is some cell lines that have a lot of genes defined as differentially expressed or highly expressed in that cell line. And then this is the distribution of the length of those gene sets. This is the red bars. Also, with the kinome scan, we can see that there is more of a hard cutoff of a certain number of kinases, or it's never really larger than the bins that we have. So there's less library, there is less gene sets. There is only 71 gene sets, and they're all short. So this is visualized with this histogram. So in the histogram, you can specify the colors and the labels of the legends, and there's many other features that you can use. This is a quick way to create scatter plots. If you use Excel to create scatter plots, it's like pretty much a pain. But here you have a lot more control and it's easier. So Seaborn is a library that actually is built on top of Matplotlib, and it has nicer graphical features. This is an example of a scatter plot heat map used in Seaborn. The plots in general, they look more like publication ready. So Ido created some heat maps with Seaborn. I'm sure you've seen heat maps in publications. And we will go over some details about hierarchical clustering and how those heat maps are generated. But in general, what you can do, you can just take distance matrix that has the values of the Jacquard indexes that connect the drugs and the cell lines, and you can visualize those as a heat map. So this is a Seaborn heat map function. On the right, you get a dendrogram of the same heat map. So on the left, you can see that it's not clustered and everything is by the order we enter the data. But once we do the cluster map, we have also the clustering of the rows and the columns. So the next thing that we can do now is to find the top drugs, cell line associations. So you can use the Jacquard Stack command, sort the values by their ascending order, so the largest to the smallest, and then find the drug cell line combinations that have the highest value, the highest overlap of the genes. So those drugs potentially are, they have kinases, are inhibited by the drug, and those kinases are highly expressed in those cell lines. Maybe, I'm sure you can criticize the idea of doing that, and potentially there's ways to do it better, but what we can do, with this command, we can just get the shaft from the hay and get the highly likely predictions for drugs that could be used for a specific cancer type that is depicted in that cell type. And we can validate whether those predictions or those associations that we identified, so those are not necessarily predictions, those are associations that we identified. We can validate them with some external data. And like I mentioned in the beginning, there is a data sets that profiled those cancer cell lines with drugs, with a list of drugs. So basically those are efforts, and one of them is called a CTD square. And this is an effort that is sponsored by the NCI, where they take those 1,000 cell lines, and then they have thousands of drugs. And then they have those experiments, high throughput experiments, to see which drugs kill or inhibit the proliferation of those specific cell lines to map drugs for cancer types. So we can see if our effort was just taking the CCLE gene expression and kinase inhibitor data without looking at cell viability data, and we can see if we are able to match it. So one thing to do, which we are not going to do in this particular lecture or class, is to look at this data set and see how much it validates our associations. Our lab also worked with the Sorger lab during the Lynx time to develop this website that visualizes cell viability of 6 breast cancer cell lines, where those 6 breast cancer cell lines were treated with about 200 drugs, mostly kinase inhibitors. And then there is also gene expression data. So the points on this plot, and you can check it in this URL, are specific signatures, gene expression signatures for a drug that is treated at a specific concentration. Then gene expression was measured at a specific time point, here, for example, 24 hours. We visualized the data based on the similarity of those signatures, and then also the coloring, the red and blue coloring is how much the cells either increase or decrease in their proliferation. So this is a combination of expression data and cell viability data visualized using a specific dimensionality reduction data visualization method that you will learn from Alex in the next few lectures about how those dimensionality reductions figures are generated. So I just want to bring up that you can start layering all of those data sets and visualize them in this way. So here, what Ido did, he went to the literature and he was looking for evidence of some of those top drugs that we ranked, and having evidence in single publications that show that some of those drugs were shown to have an effect on those particular cell lines. So this is another way to validate predictions. When you cross 2 data sets, you can go to the literature and you can see whether somebody already discovered these associations more through trial and error in low throughput experiments. So just summarizing this part of the lecture, we set up a matplotlib and Seaborn and generated some histogram and heat maps using the gene set libraries and distance matrices. And then we found the top drugs. And then we discussed a little bit about how to, once you identify those top drugs or rank those top drug cell line associations, how can you validate those predictions with additional data? But this is just touching the surface, obviously, of data visualization, plotting, and discovery methods. We will have more of that throughout the course. But again, obviously, you can think about your own research and how potentially you can apply that to your projects. So this is some more of the resources that are available that we collected for you. Another assay that I should mention is called Prism. It was also developed at the Broad, and this is a high throughput method to look at cell viability. So they basically make drugs and cells, and they were able, using barcoding, to identify which cells are sensitive to which drugs. So that's something to look into, also to validate those type of exercises of trying to find drugs to kill specific cancer cells.